this is the titles of my topic. So if you go to the next slide, please. Next, please. Okay, so I'm going to cover during my presentation and to give you overview and background about MERS, as well as epidemiology, symptoms and transmissions, and also about the vaccine and the treatment. Then we'll focus more about our experience in the experimental work using the clinical sample from patients who's died or survived with MERS-CoV. Next, please. So MERS-CoV is a novel coronavirus, positive sense, single-stranded RNA virus of the genus beta coronavirus. It was first discovered and reported in 2012 in Saudi Arabia. And the virus was isolated from sputum sample from a patient who fell ill in a 2012 outbreak of a new flu. So it appears that some people became infected after contact with camels, and according to WHO, MERS-CoV is a zoonotic virus. Next, please. So regarding the epidemiology, so far, all cases have been, epidemiological linked to 12 countries, starting from Saudi Arabia and Bahrain, Qatar, and other Middle East countries. And there are another 15 countries who have reported travel-associated cases. In terms of 2021, and the total cases so far, we have 2,578 laboratory confirmed cases, and about 188 cases with related to died patient, and fatality rate is 34.3%. However, excuse me, the majority of cases were reported from Saudi Arabia, and the number or the total cases, we have 2,178 cases. And the death cases is about 110. So you could see the fatality rate in Saudi Arabia with about 37.2%. Next, please. So what you can see here in this slide is an overview regarding the cases that reported in Saudi Arabia, as well as reported in other Middle East countries. So what you can see in the right up, you could see the number of cases that reported in Saudi Arabia, and the number of or the name of cities. So in the one in the middle, these cases reported in other countries. The other thing that to really highlight is the number. As you can see here in Saudi Arabia, we have about 110 cases reported died, and we have the highest number in terms of number of cases. I know this slide is very small. If you need more details and to have more information about cases, you could go to the WHO website. You will see this as, graphs and in more details, and you can have it clearly, and what's the update so far in terms of the cases. But the summary of this slide is that we have very small number. 2021, we have reported just 13 cases, positive cases of MERS. So it's really clearly that number of cases, it's go very, very low, and we don't have much cases increase it nowadays, especially with the SARS pandemic. Next, please. So in terms of symptoms, the clinical spectrum of MERS-CoV infection ranges from symptoms or mild respiratory symptoms to severe acute respiratory disease and death. However, the most common symptoms is that we have fever, shortness of breath, cough, vomiting, diarrhea, sore throat, and pneumonia. Those the most common symptoms with MERS-CoV patients. Next, please. So this slide is to show the MERS transmission and how what we know so far and what is that we are not certain about the transmission. What you can see here in the solid arrows, those is the confirmed pathways, and there is evidence of the transmission. So what you can see here from camel to camel is confirmed transmission. Also from camel to human, also it is confirmed. From human to household or healthcare worker, also it's confirmed. Whereas the dashed arrows, there is no supportive or limited evidence to confirm the transmission, for example, from bat to camels or from human to camel. Next, please. Okay, so there are two pathway or two way of transmissions. We have a primary and we have a secondary transmission. The primary transmission does not result from contact with a human MERS case. So it is a zoonotic transmission from camels. And there is no evidence of other sources. Whereas the secondary transmission results from contact with a human MERS cases, as I said, healthcare worker and household associated. Next, please. So here also another more details about the human and the transmission from human to human. There's from human to human transmissions, it follows zoonotic transmission. And also human are considered terminal or transient hosts only. Also what you can see here in this map, where these cases reported or where the reservoir, let's say, or the middle, I would say, host of the transmission. You could see in the red, like for example, in Middle East, where the camel and the human, where like in Africa, camel to camels, whereas in the blue, from human to human. Next, please. Here more details about transmission, what I discussed in the previous slides. What we know and what we don't know about transmissions. Those in red arrows is confirmed and there is evidence of transmission. But from those in blue, it's still a question mark, and we don't have much evidence and much supportive data to confirm the transmission from bat to human or from bat to camel. Next, please. Okay, the environmental transmission has been isolated from bed sheet, also from IV fluid, and also from X-ray devices. Also, which is suggesting that the potential of environmental transmission is needed, and there is evidence of the virus in those devices or these surface. RNA also being identified in air sample from hospital rooms of MERS patients, and there is no epidemiological evidence to definitely implicate vomit or aerosol transmission. Also very important here to highlight that the RNA is live, and it is found in lower and upper respiratory tract system. And also very important is the high RNA levels was reported and found in lower respiratory system, and more severely ill about the PCR indicate the high CT value. So that's what regarding the virus and viral load. Next, please. What we know about vaccine and treatment in terms of now 9 years since MERS-CoV discovered, there is still no vaccines. However, there are some clinical trials ongoing, some of them in phase 1, phase 2. And also there is a vaccine for camels is available. In terms of treatment, there is no specific antiviral treatment recommended for MERS infection. However, current medical is supportive and based on patients' clinical condition, for example, oxygen therapy. Next, please. Here, this slide is to summarize the vaccines candidate so far. So there are 4 ongoing clinical trial in those types of vaccines. The first one is really highlighted, and it is, in a collaboration between Saudi Arabia and Oxford University. And this paper to summarize the update so far about the vaccine clinical trial, and it is just published 2 weeks back. And for more details, you may search for it and have more details about it. The other candidate vaccine is still in phase 1, and some of them in phase 2, and from different countries and different research centers. Next, please. The very important here to highlight here is about the metagenomic and our research using clinical samples from patients. So what I'm highlighted here is the objectives for our work and what we did so far. So our main objectives in our work is to investigate the genome mutations, frequency, and viral mRNA as to accumulation in MERS-CoV infected samples, and also to identify unknown co-infection or pathogens that might present it with MERS-CoV and might increase the severity of the patients. So it is very important to study and to sequence these samples to find, is there any other microbial or any other viral or bacterial that might make or made the patient very ill, not because MERS, but there are other or secondary infections. As what is the research significant is that to address the knowledge gap by identifying the host, also host gene, classified with a patient that will act as a biomarker that can be predict the severity of MERS coronavirus infection, as well as the potential targets that the virus uses to enhance the infection will be identified. And it's really helped to have the more treatment and to know about the symptoms and to link the symptoms with the clinical patient. Next, please. So here about the study design, what we did so far, we recruit 120 patients positive MERS, and we also include 30 negative sample as well. And we the sample being collected from nasal pharyngeal aspirate, and we do using the Oxford Nanopore platforms. And that's what you can see here, the workflow about the sequence and what exactly we did in terms of extraction and do the RNA sequence and do the PCR library and do the analysis and the bioinformatics. Next, please. Next, please. This slide to show the map of Saudi Arabia and where exactly these sample collected. So you could see those in red and yellow, and the number that are available sample from patient from our biobank, and we do the sequence to see is there any difference with in terms of geographic or any difference between human genome or whatever regarding the positive samples, or if they are like more death in one region is something related with genetics or related with camels or the culture, or what is the farms, or is there any connection between farms and patients. Next, please. Here about our primer design, we use the Amplicon approach, and we, as you can see here in A and B and C, where the 30 Amplicons to cover the whole genome size of the MERS, and also the second one is about 15 size or 15 primers, whereas the bottom one is about the primer. We use 8 Amplicon or 8 primers that cover the whole cover of the whole sequence or of the genome of the MERS, so as indicated. So next, please. Here, since we have these samples and some of them collected from 2018, 2019, we need to screen them and to choose the best sample that need them for our sequence approach. So what you can see here in the left, we use 2 primers, S and N, as a screening or targeted. What is there, which means it is pass the test or pass the QC to go for the next experiment and to do the sequence. And in the right side, what you can see here is the location and just an example, what is the PCR in terms of gene and RDRB. Next, please. What you can see here is the patient characteristic and the information in terms of that been used in this study. We use just 15 or 16 sample here, those who pass the QC, and is really we have a good quality of sample to do the sequence. We did some sequence with the rest of the 120, but some of those samples couldn't cover the whole coverage or the whole genome sequence. So we those 15 is really excellent, and we have the information in terms of CT value, in terms of E gene and RDRB, and also the comorbidity, the outcome, and ICU status. Next, please. Here about the agarose gel electrophoresis, what's the amplicon that generated from 30 in A, 15 amplicon in B, and 8 amplicon combination in combination of primer pairs, which is in C, we use 8 amplicons. Next, please. So what you can see here is the red depth analysis that as a bioinformatics we follow, and we could easy to see the 30 amplicon in A and 15 amplicon in B. Those is represent the 20x coverage, indicated that the above of this lines line each nucleotide was sequenced at least 20 times. Next, please. Here about the summary of about bioinformatics pipelines and what we follow and how we analyze, and which bioinformatic tools to use in terms of sequence and in terms of analyzing and to validate our data. Next, please. Here, very interesting slide to see what is the other co-infection that with our patient samples, and if there is any indication or any link between patient who survive and with MERS and with patient who died with MERS. What you can see here is really insight about a potential causative agent for a bacterial pneumonia if one was to emerge, and we have about 20 species characterized into genus from 15 patients with severe MERS infection. Also, the human reads were removed from the sequencing library, and viral and bacterial transcripts were identified using Karrie 2. Next, please. Here also, in terms of survival and died patient, what you can see here is the up and down regulated abundance of bacteria in fatal cases of MERS CoV infection in comparison to non-fatal cases. So you could see the numbers of or the identified bacteria between those two categories. Next. So in summary, this research provided an unparalleled and amazing step forward for our health institute and system to release or to using and utilizing benign sequencing, particularly as a rapid diagnostic platform and as like real time sequencing to know what's going on. And also to understand the pathology of MERS, particularly in how viral load and which co-infection influence outcome and the severity of this. Finally, identifying the suite of host gene classifier and the sequencing approaches was used to identify cellular host mRNA whose changes in abundance directly correlate with the severity of infection and outcome of the patient. Next, please. Here for more details regarding our approach regarding this metagenomic. For more information, please, you could find it, it's published in Hemisphere, where more details about this technology and about the work that we use. Also, it's really important to highlight our approach and experience with MERS. We apply it in SARS-CoV, which exactly the same, coronavirus. So with the experience that we use and how we react and do the preparedness and the preventive with coronavirus, we apply it in South, and luckily now we have very low cases in Saudi Arabia in terms of how we can tackle these viruses, and we could apply it in somewhere, and if any other future like a pandemic or outbreak that might happen. Next slide. With this, I would like to thank our collaborative and our organization from FDA, from King Fahd Medical City, and also from other institutes who support this project and provide samples. Next slide. Thank you so much for your attention. If you have any question.
Okay, thank you very much, Dr. Waleed, for the excellent presentation. Let me check. We don't have any question from the floor. So in interest of time, just ask you one question. Based on your experience, we almost forget about MERS coronavirus because of the pandemic, and we see that the current pandemic showed that the potential of coronavirus to mutate. Even though literature says that the rate of mutation is lower than influenza, but we see that it has mutated significantly. So what do you see with regard to MERS coronavirus? Do you foresee a significant mutation to the genes?
So far, we couldn't see any more or significant mutations. What we noticed really in terms of pandemic with, SARS pandemic, we thought there is something really will be not a trouble, but we could see maybe two mix of virus at the same time. So we did really more screening on MERS. As you said, people, they might forget about MERS, but we couldn't see any positive or increased cases in Saudi Arabia. So what we also another approach we use, if we have the sign of pneumonia or coronavirus, we did first the priority. So the priority is SARS. If it is negative, then we go and screen for MERS and maybe other influenza or other viruses. So that's what in terms of approach or protocol to diagnose coronavirus. But in terms of mutation, the virus as it is, we couldn't see any mutation on MERS.
So it's fairly stable. All right, so thank you very much, Dr. Waleed, for that presentation.